I'm Brett Pickett. I'm an assistant professor here in the micromolecular biology department, and I grew up in Sandy, Utah. My research involves two different aspects. The first is comparative genomics of human pathogens, and the second is looking at how human cells respond during infection with those pathogens. One of the examples that we're looking at right now is comparative genomics of influenza virus and how it changes over time. So we're using computational methods on publicly available data to find those trends using statistics and then figure out exactly what that means. The second aspect is during infection, we're using high throughput data and big data methods on the supercomputer here on campus to try to put all those pieces of sequence back together and quantify them to measure how the human cell is responding. On the influenza side, we're looking at how and why the virus mutates and how that affects disease. So we're using statistical methods to try to make sense of where it's mutating in the viral genome and then what phenotype that change can take. On the transcriptomic side and the human response during infection, we're using the supercomputer to put together all the different sequencing reads and measure how each mRNA goes up or goes down during infection. Then we're mapping that into which pathway, which signaling pathway that those genes belong to, and ultimately try to find a drug that can affect that pathway and reduce replication of the pathogen. My undergraduate work was here at BYU. I majored in microbiology with a minor in Spanish. And from there I went on to a PhD program at the University of Alabama, Birmingham. From there went to Texas, so the UT Southwestern Medical Center in Dallas. Did some postdoctoral work in the pathology department. Then we moved to San Diego. I had a couple of different research projects in the nonprofit space and also in the for-profit sector. So applying both comparative genomics methods and also transcriptomic methods and identifying potential drug targets for repurposing existing drugs to a new therapeutic area. I came to BYU for the academic environment this year and the ability that it has to combine the academics with the gospel-centered learning. I think that's a unique aspect of BYU and a big draw to why I'm here. I'm excited to teach students not only in the classroom but also in the lab how to apply these methods to their own individual research project and how we can make sense of the results that come out of that and ultimately advance scientific knowledge. I think anybody that's looking into MMBIO major should do it. There's a lot of interesting things being taught in the classroom, from how DNA works to how it's made into protein to even how germs replicate inside our bodies and how our immune system fights it off. I think a lot of the skills that we teach, a lot of the knowledge that we teach in the classroom is applicable to jobs outside of school, whether it's in academic research or as a technician in a lab or even as a researcher in the industry. I think there's a lot of demand for people in MMBio, and a degree in it would serve any student very well in the future.